Okay, everybody. Hope you can hear me all right. Welcome back. Welcome to session 3 of our 2025 Open Research Conference. I'm Lucy May. I'm the Open Research Manager here in the Office for Open Research at Manchester. And I'm delighted to welcome you to this session of talks, which are focused on open research in collaboration. So we've got two talks for you on this important topic. We're first of all going to hear from Dr. Michael Biddle, joining us from the University of Leicester. He's going to be talking about an open collaboration between industry and academic partners. And then we're going to hear from Professor Ellen Poliakoff, who's one of our open research fellows right here at Manchester. And she's going to be talking about some public contributions and participation in open research and the questions that raises. So I think you know the format by now. It's 15-minute talks from our speakers and then about 5 minutes for some questions. And then we'll be moving into a panel discussion to end the formal part of our day today. So delighted to welcome Dr. Michael Biddle. Thank you.
Okay, so hello, I'm Dr. Michael Biddle. Today I'd like to talk to you about improving biomedical research through an industry and academic open research collaboration. So to start off, I'd like to point out why improvements in biomedical research is important, and one of those is actually drug development. So currently drug development, it can be slow, expensive and risky. There's various barriers to cause this, such as absence of poorly sensitive or specific tools. So if you're interested in a given protein, which may be a potential therapeutic, not being able to detect that protein from the get-go is quite a big barrier. It could also be poorly predictive model systems. So let's say you've got a drug that looks really promising in a mouse model, that may not necessarily translate to humans. Or there could be uncharacterized patient heterogeneity. An example of this is, for instance, imagine the asthmatic population. Some of these patients may respond to the treatment and some don't. And if you can't define that population, it's hard to know what's causing the lack of effect. But one major barrier, which is kind of not spoken about, is actually irreproducibility and lack of quality within biomedical research. Now, these are all publications where they've tried to reproduce the main finding of publications from biomedical research. And what they find is on average around 50% of papers are irreproducible. Now, if you look at the US annual spend, that actually accounts for $28.2 billion per year in terms of waste. And one of the main factors what causes this is biological reagents and reference materials, and antibodies are one of the most common biological reagents used by biomedical researchers. Now, just to summarize what an antibody is for those who don't know, it's a Y-shaped protein, and they're used in biomedical research, they're used to understand the involvement of proteins in disease and mechanisms, but it can also be used for therapeutics or diagnostics, for instance, quantifying proteins within the blood to suggest treatments. Now, a good antibody on the right, as you can see, that's a very basic picture of a cell, should be detecting the given target protein, which here is resembled by the green rectangle. But some antibodies, it turns out, are non-selective, and they also detect other proteins, which isn't ideal. And some antibodies don't even detect the target protein at all. Now, to give you an idea of why this is a huge problem in biomedical research, this is just an example from my PhD. So NOX4 is meant to be a wonder protein, where if you treat that, you've cured most diseases. So on my PhD, I started using the most used antibody in the literature. And what we found was really nice staining on the left on cells called airway smooth muscle cells, which are one of the main effector cells in asthma. So I was like, great, this looks really promising. And then I thought, well, it's probably best to check that my data is reproducible. So I actually went away and had to check the antibody. And this is a long process, which involves you extract the gene for that given protein, put it into a circular piece of DNA, what we call a plasmid, and introduce that into cells, which should increase the protein of interest, which allows you then to test your antibody against it. You then you can do multiple techniques. The technique of interest at the time for me was Western blot. And what happens is you load your protein sample from those cells here, and it travels, you apply an electric charge, and then your protein appears in the gel. We then transfer that onto a piece of, it's essentially just a membrane, a piece of paper called a nitrocellulose membrane to allow us to detect the protein with a given antibody. Now, if an antibody works, what we should see is the cells which have been introduced with that NOX4 DNA should increase, and the control cells should have a very thin band. And if the antibody's failed, there shouldn't be an increase. And what I found is the most used antibody in the literature was rubbish. And since then, it's been discontinued, but has misled the field completely. So as a researcher, the next thing, what you should do is go on to something like this, which shows you all the antibodies for NOX4. And it turns out not many people are aware this exists. And on this platform alone, it told me there's 952 antibodies for NOX4. This is a massive problem, though, because antibodies are resold between suppliers. So you could be buying the same antibody 4 times without knowing. Then the question is, well, which ones should we pick? Well, citations that are just shown don't mean necessarily a good antibody. Is there any data to validate the antibody? And for a lot of these antibodies which aren't heavily researched in the field, the answer is usually no. And you could go on, manufacturers' recommended techniques, but if they've not validated it, then also it's meaningless. In the end, I checked 12 antibodies, which costs around £4,000. So for a PhD student, that's just not feasible, in my opinion. We tested them on a machine called a Tune Next, which is basically a flow cytometer. But for basically what we should be seeing is this blue histogram shifting to the right if it worked. And it turned out 1 out of 12 worked. So £3,600 wasted. And if you've only got a bursary of 5,000, that's essentially a year's bench fee gone for a biomedical researcher. As such, between me, Harvinder Virk, and Eva Krakow, who's a behavioral scientist, we established the Any Good Antibodies Community and Forum to try and address this problem. And this aimed to work with all stakeholders to increase the availability of high-performing antibodies and reduce the use of these poorly performing antibodies, which are misleading the field. To establish this, we've approached it with an interdisciplinary approach. We actively engage all stakeholders, and we have a no-blame culture, because I think that's one of the main barriers of why this hasn't been addressed previously. And we support open science and transparency. So over the last year, we've been establishing and getting all the stakeholders in the room to do a Delphi process, but we also wanted to have a focus group to ask people, well, what decisions are you making for antibody purchasing? And their reasons for purchasing was based on citations or reputation, which was alarming, but also expected. There's also perceived barriers in antibody validation, which was the awareness that they didn't know they needed to do it, as well as the incentive because you can publish without doing it. So they came up with awareness, education, and trusted data sharing as ways to kind of solve this. So we took away from all that there needs to be a more standardized approach for antibody validation, and we need to overcome the expense to produce this. We need to improve education to improve end user choice. And then we also need to promote the newer technology of antibodies, which we have not touched on, but I'll go on later, because people are still using the oldest technologies, which are the least reproducible. To go about this, we've done various education things. We've done a blog, we've done a report with the NC3Rs, which is the funding group, and we've also done a conference with the NC3Rs on this matter. We've done masterclasses, and we've actually got a website now, which will hopefully host even more education as time progresses. Alongside my realization that NOX4 antibodies were terrible, Carl Laflamme, who's in the bottom center there, found out that his protein of interest, C9orf72, had the same story. And this isn't just an isolated case, but Carl decided that he wanted to do something. So him and Alid Edwards and Peter McPherson decided to set up ICARUS, which stands for Antibody Characterization in Open Science. And this was set up in Montreal in Canada. And I went on an industry exchange and established an ICARUS site in the UK. So we've gone from improving education now to actually directly assessing the antibodies which are causing the problem. And how this works is funders provide us with a list of antibodies they want us to investigate. We then feed those, proteins, we then feed them to the manufacturing partners, and we only work with manufacturing partners to ensure that we're not getting the same antibody 50 times. And they provide us with antibodies they'd like us to assess, and then we disseminate this data openly with Zenodo, F1000 Research, and SciCrunch, which is the antibody depository. Just to quickly go over the data which we show in these reports, we have 4 techniques. So the first technique is a Western blot, so a good antibody should look like this. An antibody what could work but is a bit messy will have bands on the right, and to explain this further, control basically means the cells with the protein in, and then what we do is we artificially delete that protein from the cell essentially, which is a knockout. So what we should be seeing is no protein in the KO lane. But then some antibodies again don't recognize that target protein, such as A0 and 992 there. We also do immunoprecipitation, which I won't go into details with because it's a bit of a headache to explain, to be honest. But you basically what you're after is an increased density on the right here. So for instance, MAB 4364 looks like a really good antibody to detect this protein. But if it fails to immunoprecipitate, you don't see anything on the right. So this is an example of a failed one. And what you see in there, this band here, is a non-specific band from the way the system works with an alternative antibody. Immunofluorescence is a really cool technique which allows you to see where the protein is in the cell. So what we do is we stain these cells with different dyes. We combine them on a plate. So imagine that's a glass slide and we've got a mixed mosaic pattern. And what a good antibody should be is we should see the staining in the green cells, which is control cells, and no staining in the pink, like here. But as you can see from this image here, most antibodies are failing, and that's an example of a failed one. Now, in the UK, we've established the flow cytometry protocols, which is really cool because that can be used for diagnostics from clinical samples, for instance. And it's the same procedure. We have pink and green stained cells, essentially, with fluorescent dyes. They get mixed, run on this machine, and what we should be seeing is this pink histogram here overlaying with this dotted line. So good examples here, and the green should shift to the right. But on flow cytometry, like immunofluorescence just showed you, many antibodies are failing. Now, when you collectively combine all this together, it's not a good picture for antibody use in biomedical science. But the positive news is that for every protein, we usually find one or more antibodies what actually work. And we feed that back to the biomedical research communities and funders to actually disseminate that data further. But as you can see, recombinant antibodies which are there, which I mentioned earlier on, are actually performing the best, and the polyclonal and monoclonal antibodies are worse. Now, immunofluorescence actually performed the worst, and this was published in eLife. So I went away and actually looked at, well, if IF's actually one of the worst performance for antibodies selectivity, well, let's see if anyone's validated in the literature. And in biomedical research, with the 80 antibodies what resulted in the failed IF, resulted in 369 publications, but only 11.6% of them actually checked their findings. So the rest of the publications there may not be reproducible. To help people understand this data more and also to help disseminate it, because it's hard to find these things, we've actually started establishing a database which will be openly shared and openly accessible for any biomedical researcher. And the funders are actually going to use this database to also recommend antibodies for a given field. So imagine if I'm interested in Parkinson's, I can then look for my genes of interest and then go to the people who are applying for the grant and say, we recommend you to use this. Collectively, we've seen massive benefits of going through an open research thing. This platform wouldn't work without this. In terms of the manufacturing partners, they want everything to be openly shared, and it stops them also saying, well, we don't believe the data because it's all done side by side comparatively. It also allows researchers to adopt it quicker, and it also allows it to be published quicker, which is obviously massively important for the communities who are using this data. And from doing this over the last 2 years or so, we've seen that it's had a massive resultant impact. And I think that's also good to recognize with open science. So we've had consensus protocols agreed and published, and all these are available on protocols.io. We've had a new funding scheme announced by the NC3Rs to improve antibody reproducibility and reagent essentially tools to detect protein validation. And the NC3Rs are currently doing a policy review. We've actually got more than 60% of antibody manufacturers join now, and we've recently just had another one join, which is great news. So we're actually able to assess the majority of the market now. And then just to demonstrate how the benefits of open science can lead to further things as an individual, we've also been given the Dorothy Bishop Award for reproducible and open research culture by the UKRN, another impact case study published, as well as various sources of funding. And really, if you focus on better antibodies, the take home really I want people to take away from it is it results in better science. And then when you apply that to patients in particular, so let's say this protein is what may be important for disease, we can then identify good antibodies for them to make blood tests to hopefully improve patients' lives and better clinical trials. So in conclusion, better antibodies result in better science, better patient outcomes. There's a QR code if you want to get in touch or know more. And I'd like to thank all my collaborators. Thank you.
Thanks, Michael. That's fantastic. Okay, have we got any questions for Michael? Stephen's always got a question.
Really nice talk. I enjoyed all the antibody stuff since my background is in life sciences, but
Oh, that's good.
I mean, it's great to see that you're working with commercial providers, but was there any resistance from them for that? Because maybe some of your work shows that they've been selling dud reagents to the community for years and years. So
Yeah, so the pitch we provide them is it's also costly for them to keep these dud reagents going if they're rubbish. So we're like, well, we'll clean up your catalogs, and they are taking down the dud reagents, not as quickly as I'd like, but I imagine there's in-house testing they do following the data sets. The pitch also is we demonstrate gaps in the market. So for instance, parkin, the protein called parkinase, most of those antibodies were terrible. So Thermo Fisher jumped on board and released 2 really good antibodies. The advantage of that is it benefits the community, but benefits them. So when you give them those kind of pitches, obviously initially there was some, but then after that, it's just been kind of a slow adoption of the rest. So yeah.
Probably got time for one more question. Oh, right at the back. Thank you. Just bear with us a second while we bring a mic up.
Yeah, nice presentation. My question is like, you report good antibodies, right? Only good antibodies. Can we report the, the antibodies that is worst? Like my experience is like, I ordered some antibodies from, Santa Cruz and it was not working. Like I spent 6 months and no results. Then I ordered antibody from Cell Signaling and that's wonderful. Like so is there any way like, so there is a stringent rule like a regulation like a drugs. So in the antibodies as well. So we can save our precious time. Yeah, thank you.
Yeah, so to give you an example, we screen on average 12 antibodies, but that can range up to 30 depending on the target. If the antibody doesn't perform well, we feed that back to the industry manufacturing partners, but they can't say, don't publish that. That still gets published. It's more so they can have an opportunity to say, well, have you tried this in a scientific manner? They can't say, don't publish it because we don't like the data. That's the agreement we have. So all the bad antibodies still go out and we show that. And then on the database, we'll have, the bad antibodies as well as the good antibodies. So you know which ones are worth getting and which ones, from an individual perspective, you may not want to get.